Hello guys, welcome back to my audio space. Hope you all are doing and learning well. So today we are going to look at the structure of a eukaryotic cell. If you wish to know about the structure, function and reproduction in prokaryotic cell, please go back and watch out videos on prokaryotic cell. So without taking no more time, let's get started. Yes, so before getting deep in the structure of an eukaryotic cell, let's first discuss about why eukaryotic cell is considered as more advanced and complex than that of a prokaryotic cell. Here you can see on the left hand side, the cell is having well-defined nucleus and so many embedded structures which is membrane bound, this structure, okay. So it looks much complex, and if you look on the other hand, here no well-defined nucleus is present at the center, and on the name of embedded structure we can see over here these are small dotted like structure called ribosomes. This cell is very simple in structure, so this complex structure of cell is eukaryotic cell and the simple cell is prokaryotic cell, as I already told you. So what is an eukaryotic cell? The word is taken from the Greek word where suffix "u" means well or good, okay, and karyon means nucleus. The cell having well-defined nucleus is known as a eukaryotic cell. So, an eukaryotic cell contains a membrane-bound nucleus and other membrane-bound structures known as cell organelles, which are absent in prokaryotic cell. Basically, a cell consists of three essential parts. What are these? These are cell membrane, cytoplasm, and nucleus. If we draw a structure of a cell, you can see this is the outermost covering, which is cell membrane or plasma membrane. Inside this, there is a semi-liquid or colorless substance is present, which is known as cytoplasm, where all the cell organelles are embedded, and inside which there is a nucleus, which is spherical in shape that contains genetic material DNA. Okay. What is cell organelles? There are many membrane-bound structures present in the cytoplasm which perform specific functions. These are known as cell organelles. So, there are some cell organelles which are membrane-bound structures are present in the cytoplasm. They are known as cell organelles, which are almost completely absent in the case of prokaryotic cell. Eukaryotic cells are present in animals, plants, protists, and fungi. Now, let's discuss about all the components of a eukaryotic cell one by one. The first one is cell membrane. It is also known as plasma membrane. Basically, it separates the interior of the cell from the external environment. Let me draw a cell. Suppose this is the cell membrane. What it does, it separates the outer surrounding to the interior of the cell. Structurally, it is composed of phospholipid molecules. So what are these phospholipid molecules? The molecule consists of two parts. The head and a tail. Okay. So The head having phosphate group, and which is hydrophilic. Hydrophilic means hydro means water and philic means loving. This is hydrophilic. So it always pointed toward the water end or liquid end. Okay. Hydrophilic. This is hydrophilic, and the tail are made up of two fatty acids which are hydrophobic in nature. Hydrophobic means what? Hydro means water and phobic means to hate. So it always pointed away from the water medium. So if we magnify this particular portion, then what we see? See here. This is the structure of plasma membrane. It is composed of phospholipid molecules. This is phospholipid molecules, the head, and these are the tail which are pointing inward. So basically two layers are required in the formation of plasma membrane. So this is known as phospholipid bilayer. Okay. And it is also, you can see this structure. What are these? These pink bodies. What are these? These are proteins. These are embedded proteins. So basically two types of embedded proteins are present in plasma membrane. These are, someone are completely inserted in the plasma membrane and some are loosely attached. So the integral protein, this span the membrane and help in transporting molecules, and the peripheral protein, what they do? They are loosely attached and help in cell signaling or act as enzymes. Okay, so it helps in transport of molecules, cell signaling and protection. Next one is cytoplasm. Inside the cell, wait, what is going on? So this is a cell, this is the nucleus, and this outer layer is plasma membrane. So inside the cell, this area is filled with some sort of fluid which is semi-liquid and colorless, which enclosed by cell membrane. This semi-liquid material is known as cytoplasm. All the cell organelles or components of the cell are embedded in it, and it provides protection from damage. It provides cushion to them, thereby protects them from any kind of damage. It helps to store the molecules which are responsible for cellular processes and is also help in providing shape of the cell. So the next one is nucleus. Suppose if this is a cell and at the center of this is the nucleus, which is spherical body located at the center of the cell in case of animal cell. It is thought to be the largest organelle of the cell. Inside this cell, okay, so this is the structure of a nucleus. You can see over here, this is the enveloped structure. Inside this, it is filled up with some sort of liquid known as nucleoplasm, which is enveloped by two membrane covering known as nuclear membrane. So this is double membrane structure which surrounds the nucleoplasm, or we can say the nucleus, which is fenestrated by small pores known as nuclear pores. As I just told you, nucleus is double membrane structure. The inner membrane, this one which facing towards the nucleoplasm, is inner membrane, and the outer membrane which faces toward the cytoplasm. This is the cytoplasm and this is the nucleoplasm, and this complete structure is nucleus. The outer membrane of the nucleus, known as outer envelope, is continuous with endoplasmic reticulum that we will discuss later. Inside this nucleus, there is a genetic material is present. Genetic material or DNA is present, which houses the genetic information, thus responsible for heredity. It plays also important role in cell division. So the next one is nucleolus. You can see over here, inside the nucleus, a dense region is present, which is shown by this violet color. This region is known as nucleolus. Nucleolus is responsible for producing and assembling the ribosomes. After assembly, the ribosomes are transmitted to the cell cytoplasm, where they serve as the sites for protein synthesis. So what? This nucleolus is responsible for the formation of ribosomes. Once the ribosomes are formed, they are then transported through these nuclear pores to the cytoplasm of cell. So the next one is endoplasmic reticulum. In this structure, you can see over here, this is the nucleus, okay, having inner nuclear membrane and outer nuclear membrane. The outer nuclear membrane is continuous with this irregular sheets or stacks of double membrane structures, which are known as endoplasmic reticulum. These are series of irregular networks made up from double membrane structures. So at the inner end, it is connected with the nuclear membrane, and at the outer end, it is connected with the cell membrane, thereby forms the supporting framework of the cell. Like this. If this is a cell and this is a nucleus, so the outer nuclear envelope is continuous with this irregular sheets. Okay. So at the inner end of this endoplasmic reticulum, it is connected with what? It is connected with nucleus, and at the other end, it is connected with the plasma membrane or cell membrane. So it acts like a supporting framework of the cell. Functionally, it helps in distribution of material from one end to another end. Now, there are two types of endoplasmic reticulum found. So, in the context of two forms of endoplasmic reticulum, the first one is rough endoplasmic reticulum, or you can say RER. In this picture, you can see the difference between this purple structure and this yellow structure. These pink color having dots all over. See, there are dots all over. Due to these dots, it looks like rough or granular in appearance. So this one is known as rough endoplasmic reticulum. These dots are nothing else than ribosomes that are coming out from the nucleolus. So which are just next to the nucleus, contain ribosomes embedded in the cisternae. What is cisternae? These projections are known as cisternae. This is rough endoplasmic reticulum. It gives granular appearance, that's why also known as granular endoplasmic reticulum. Basically, RAR is responsible for processing and transporting proteins synthesized by ribosomes embedded in it. So, what is the function of RAR? The function of RAR is to processing and transporting the proteins which is synthesized by these small dots or ribosomes that are embedded in their membrane. So, the second form of endoplasmic reticulum is smooth endoplasmic reticulum. Okay, so this structure just after the rough endoplasmic reticulum, which looks quite smooth because of the absence of these dots or granules. Therefore, those cisternae which are lacking ribosomes are smooth endoplasmic reticulum or SER. So those cisternae which are lacking embedded ribosomes are smooth in structure. Therefore, these are called smooth endoplasmic reticulum. Functionally, it is responsible for lipid synthesis, formation of some steroids and detoxifying drugs. As there is no ribosomes are present, so they are absent in the processing of proteins. So we were just talked about the granules structure on the surface of endoplasmic reticulum. So what are these structures? What are ribosomes? Basically, ribosomes occurs in two forms. First one is free ribosomes that presents freely in the cytoplasm, and the other one is that are embedded in the cisternae of endoplasmic reticulum. Basically, it is made up of proteins and RNA. So ribosome structure is completely made up of protein and RNA. RNA stands for ribonucleic acid. Structurally, it is composed of two subunits. The first unit is large unit and the second unit is small subunit. In the case of eukaryotic cell, the large subunit is 60S and the small subunit is 40S, where the S stand for Swedberg unit. These are responsible for protein synthesis, therefore also known as factories for protein synthesis. So the next one is Golgi apparatus or Golgi bodies. If we look at the picture, it's having two phases, the cis phase and the trans phase. The cis phase where incoming of proteins in the form of incoming transport vesicles takes place. It's having multiple stacks, like you can see these are the multiple stacks known as cisternae, where incoming proteins are modified and sought out. So when the incoming proteins are get inside these cisternae, what these cisternae does, these are help in modifying and sorting out the proteins. After that, from trans phase, okay, from trans phase, the modified and sorted proteins get secreted out in the form of secretory vesicles. So these are the series of membrane bound sacs which is originated from the endoplasmic reticulum. Functionally, it helps in packaging, sorting and modifying proteins before they get secreted out by the cell. The proteins leaving the Golgi bodies travels in secretory vesicle. So what is the main function of the Golgi apparatus? I hope you understand. Basically, what it does, the unsorted or unmodified proteins get entered through the incoming transport vesicles. Then inside these cisternae, these are modified and sorted out, and then they get secreted out in the form of secretory vesicle through transphase. So the next one is mitochondria. These are the spherical, thread-like or rod-shaped double membrane structures. You can see over here in this picture, this is the mitochondria. Okay, this is the structure of mitochondria, which is consist of two membrane. So the mitochondria is double membranous structure. The inner membrane, this one is the inner membrane and the outer membrane. It is composed of two membrane, the inner membrane and the outer membrane. And the space between these two membrane is known as intermembrane space. The inner membrane form, you can see this inner membrane, these forms some invaginations toward the inner side of the mitochondria. These finger-like processes that are basically increases the surface area of inner membrane where FOF1 complex are present. So these finger-like processes increases the surface area of inner membrane where these FOF1 complexes are present, which are helpful in cellular respiration. Thereby, they increases the area of cell respiration. So, let me read out this. It is surrounded by an outer membrane and inner membrane. The inner membrane form finger-like processes known as cristae. Okay. Their size and number varies according to the requirement of energy. So their number and sizes varies according to the requirement of energy. If the cell required more energy, then the number of mitochondria increases in that cell, and if that particular organ require less energy, then the number of mitochondria greatly reduces. So this is site for cell respiration. It is known as powerhouse of the cell. So what is the which cell organelle is known as the powerhouse of the cell? The answer is mitochondria. It breaks down the molecule and produce energy in the form of ATP, adenosine triphosphate, which is used by the cell for performing many metabolic processes. So the next cell organelle, which is very important, is lysosomes. They are group of specialized vesicles that contain hydrolytic enzymes that help in break down unwanted molecules. So look at the structure. It's having very simple structure. These are spheres type made up of lipid bilayer. So this is lipid bilayer which encloses a fluid that contain hydrolytic enzyme mixture. They carry out intracellular digestion by destroying and digesting foreign substances. So intracellular means within the cell. Within the cell, if they find any unwanted molecule, they used to digest it by help of hydrolytic enzymes. They act as digestive bags as they digest stored food during starvation. They are also known as suicidal bags as they disintegrate worn out organelles and damaged cells. So the cell organelles which are not in their function, these suicidal bags helps to disintegrate that. So the next one is centriole. Basically, they are involved in organizing microtubule in the cytoplasm. In the cell, if this is the nucleus, then there is a clear region present which is known as centriole. It is located near the proximity of the nucleus. Okay, this clear or dense region is known as centriole, which is located in close proximity to the nucleus. Functionally, it helps in the formation of spindle fiber during cell division. Due to these spindle fibers, the chromosomes get separated. So basically this centriole or centrosomes helps in the formation of spindle fiber, and in this spindle fiber, when the chromosomes get replicated, they are separated out like this. They are separated out. Suppose these are the spindle fibers. So the chromosomes get, these are the chromosomes. They get attached to the spindle fiber, and with the movement they get separated during cell division. So this centriole is found only in animal cell. This is the clear area of the cytoplasm near the nucleus that we just learned about it. These microtubules assist in spindle formation during cell division. Next one is peroxisomes. So these are specialized vesicles that help in the formation of hydrogen peroxide, that's H2O2. Structurally, it is composed of lipid bilayer and having a central crystalline core. Functionally, they help in breakdown of fats and detoxifying dangerous substances, especially in the liver. Next one is flagellum. In some eukaryotic cell, like in prokaryotic cell, a flagellum is present, which help in motility of the cell, as we already discussed in case of prokaryotic cell. So in some eukaryotic cell, this flagellum is also present, which is made up of microtubules. The arrangement of microtubule you can see over here, these are arranged in this form. Okay, these are the central microtubule and these are attached to each other by dyneem arms, and structurally at the outside they are enveloped by plasma membrane, or we can say these are the extensions of plasma membranes inside this, which are embedded structure known as basal body. Functionally, the eukaryotic flagellum is served in cell motility, sensory and transport functions. Thanks for watching.